Untuk semua hadirin, pastikan anda telah scan My Sejahtera yang berada di luar bilik ni kan? Ya. Mungkin untuk kalau ada KKM datang nanti kita tanya. Sudah scan My Sejahtera? Ya, My Sejahtera tu berada di luar pintu ya bagi yang belum scan. Dr. Rasid Mail, Timbalan Naib Chancellor, Akademik dan Antarabangsa, Universiti Malaysia Sabah. Hadirin mohon berdiri. Nyanyian lagu Negaraku dan lagu Sabah Tanah Airku. Hadirin dipersilakan duduk. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh dan salam sejahtera. Yang berbahagia, Profesor Dr. Rasid Mail, Timbalan Naib Chancellor, Akademik dan Antarabangsa, Universiti Malaysia Sabah. Yang berbahagia, Profesor Dr. Charles Vairappan, Pemidato Syarahan Perdana. Yang berusaha, Profesor Madya Dr. Monica Sulaiman, Pengarah Institut Biologi Tropika dan Pemuliharaan, pegawai-pegawai utama dan kanan UMS, div kehormat, hadirin dan hadirat sekalian. Bertemu kita pada pagi ini secara langsung dari Universiti Malaysia Sabah dalam Majlis Syarahan Perdana yang berbahagia Profesor Dr. Charles Vairappan. Terima kasih kepada semua yang bersama-sama kita pada pagi ini. Majlis ini juga dapat diikuti secara langsung melalui Facebook rasmi UMS. Jangan lupa untuk like dan share Majlis Syarahan Perdana ini. Bagi memberkati lagi majlis kita pada pagi ini, dipersilakan Ustaz Muhammad Fazri Abdul Hamid untuk memimpin bacaan doa.
Assalamualaikum warahmatullahi wabarakatuh. A'udzu billahi minasy syaithanir rajim. Bismillahirrahmanirrahim. Alhamdulillahi rabbil alamin. Wassalatu wassalamu ala sayyidina Muhammad khatamil anbiya'i wal mursalin wa ala alihi wa ashabihi ajma'in. Allahumma ya Rahman ya Rahim, yang memperkenan segala permohonan hamba-hambamu ini menadah tangan penuh ketulusan dan kerendahan hati, memohon perkenanmu, keridhaanmu dan nikmat syurgamu, dan kami memohon hindarkanlah kami daripada kemurkaan dan azab nerakamu. Allahumma ya Allah, bersempena Majlis Syarahan Perdana pada pagi ini, kami merafakkan kesyukuran di atas izinmu sehingga majlis ini dapat direalisasikan. Semoga majlis ini berjalan seperti yang dirancang, disulami dengan hati yang tenang dan diakhiri dengan penuh kasih sayang. Allahumma ya Hakim ya Azim, sesungguhnya kami berhimpun di sini bagi menyatakan kesungguhan, komitmen dan iltizam kami dengan roh dan semangat untuk membangun, membina serta mendidik dan memartabatkan anak bangsa supaya berbudi dan berjasa kepada agama, bangsa dan negara. Justru itu, berkatilah pertemuan kami, berkatilah jua usaha-usaha kami, ilhamkanlah dalam fikiran dan minda kami idea-idea yang bernas, pandangan-pandangan yang jelas dan keputusan-keputusan yang waras. Kurniakanlah kepada kami limpahan ilmu yang berguna dan kepahaman yang benar. Satukanlah hati-hati kami, eratkanlah silaturahim di kalangan kami, padukanlah tenaga dan usaha kami untuk memartabatkan pendidikan bangsa secara bersama demi mencapai kecemerlangan dan keridhaan-Mu. Allahumma ya alim ya kabir, jadikanlah kami ya Allah dari kalangan hamba-hamba-Mu yang taat dan bersyukur. Jauhilah kami dari melanggar segala perintah-Mu. Kurniakanlah apa kiranya ya Allah kepada kami kesihatan, kesejahteraan, fikiran yang waras, minda yang cergas, rezeki yang halal dan berpanjangan, zuriat dan generasi yang beriman, serta segala kebaikan di dunia pun juga di akhirat. Allahumma la sahla illa ma ja'altahu sahla. Allahumma ja'al jam'ana hada jam'an marhuma wa tafarruqana min ba'dihi tafarruqan ma'suma. Allahumma ighfir lana dhunubana wa li ikhwanina alladhina sabaquna bil iman wa la taj'al fi qulubina ghillan lil ladzina amanu rabbana innaka ra'ufur rahim. Rabbana atina fid dunya hasanah wa fil akhirati hasanah wa qina adzaban nar. Wa sallallahu ala sayyidina Muhammad wa ala alihi wa sahbihi wa baraka wa sallam. Walhamdulillahi rabbil alamin.
Amin, amin, ya Rabbal Alamin. Majlis mengucapkan terima kasih kepada Ustaz Muhammad Fazri atas bacaan doa itu tadi. Hadirin sekalian, sebagai pengenalan pemidato kita pada pagi ini, marilah kita bersama-sama menyaksikan tayangan video perjalanan akademik yang berbahagia Profesor Dr. Charles Vairappan. Sama-sama kita saksikan.
my great pleasure to say a few words on this wonderful opportunity. JICA has been working together with UMS, especially Professor Charles. We've conducted several joint collaborations, including Technical Cooperation Project, Friends of Bornesis, Third Country Training Program, and the Skeleton of the Elephant. The collaboration with UMS in general and with Professor Charles in particular have been very successful and highly evaluated not only in Sabah but also in the world. I do hope that the collaboration between EMS and JICA will be further strengthened. Thank you so much for your attention.
Ciao. This is a pleasure to congratulate you from Rennes, France, in Brittany. So we are both working on natural products, on chemical ecology, and on lichens. So it would be a pleasure to share again with you from lichens from Brittany or from Kenya Mountains. Hi, ciao.
Hello, Dr. Charles. I'm pleased to say my talks for your requests. Unfortunately, we did not see each other a long time due to the pandemic. It's quite long since we know. We are an expert in natural products, especially seaweed, to determine the chemical structure. I'm also studying on seaweed for developing bioactive material and marine pharmaceuticals. We helped each other, so you helped me for chemical structure identification. I helped you to evaluate bioactivity by using cells and the jellyfish model. Hopefully we can see again for our collaborating work and the friendship. Bye.
Hi, this is Professor Inderpal Singh from Naipur, Mohali, India. I am here to tell you today about my amazing friend and collaborator, Charles Vairappan, who has been doing great research at University Malaysia, Sabah, for last almost 21 years in the important fields of natural products chemistry, chemical ecology, and drug discovery. He has also worked on natural product-based technology transfers. We have been in interaction for almost 10 years now. I have visited his lab, worked closely with his students, and he has visited us a couple of times in the last 10 years. And I hope in future we'll continue these collaborations among ourselves between the university and Naipur and Charles and myself. I wish Charles all the best for his career at University Malaysia, Sabah. Thank you.
Demikianlah tadi video perjalanan akademik pemidato kita pada pagi ini. Tanpa membuang masa, majlis kita diteruskan lagi dengan menjemput yang berbahagia Profesor Dr. Rasid Mail, Timbalan Naib Chancellor Akademik dan Antarabangsa Universiti Malaysia Sabah untuk menyampaikan ucapan pengenalan pemidato. Majlis dengan segala hormatnya mempersilakan.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi taala wabarakatuh. Salam sejahtera. Salam UMS Kampus Rahmah. Terima kasih Puan Pengerusi Majlis yang diraihkan Profesor Dr. Charles S. Vairappan, Profesor Institut Biologi Tropika dan Pemuliharaan IBTP, pegawai-pegawai utama universiti, para tetamu kehormat, para jemputan, seluruh warga Universiti Malaysia Sabah, dan turut tidak dilupakan semua yang sedang menonton secara langsung menerusi laman web Facebook rasmi Universiti Malaysia Sabah. Terlebih dahulu, izinkan saya menyampaikan salam takzim daripada Yang Berbahagia Profesor Datuk Naib Chancellor Universiti Malaysia Sabah yang tidak dapat bersama kita pada hari ini di atas urusan dan komitmen lain di peringkat kementerian di Kuala Lumpur. Bagi maksud tersebut, izinkan saya menyampaikan ucapan pengenalan yang telah disediakan oleh pejabat Yang Berbahagia Datuk Naib Chancellor. Tuan-tuan dan puan-puan para hadirin yang dihormati sekalian, Assalamualaikum warahmatullahi taala wabarakatuh. Salam sejahtera, salam UMS Kampus Rahmah. Syukur kehadrat Allah Subhanahu wa taala kerana dengan limpah kurnianya dapat kita hadir pada majlis yang berbahagia dan bersejarah ini. Alhamdulillah, rasa syukur turut dipanjangkan kehadrat Allah Subhanahu wa taala dalam kita membiasakan diri menyusuri dan mendepani kehidupan norma baharu. Pengurusan Universiti Malaysia Sabah terus mempertingkatkan usaha dalam memartabatkan penyaluran dan perkongsian ilmu bersama-sama dengan masyarakat. Saya ingin merakamkan penghargaan dan terima kasih kepada Jawatankuasa Penganjur dan semua pihak yang terlibat sama di dalam menganjurkan acara ini, sama ada secara langsung maupun tidak langsung, bagi menjayakan majlis ilmu pada hari ini. Hadirin yang dihormati sekalian, pada pagi yang berbahagia ini kita berkesempatan untuk mengikuti perkongsian ilmu bersama tokoh ilmu UMS dalam siri Syarahan Perdana Profesor 2021. Hadirin yang dihormati sekalian, sebelum melanjutkan bicara, eloklah kita memperkenalkan pembicara syarahan perdana pada hari ini. Beliau lahir pada 4 November 1967 di Georgetown, Pulau Pinang, dan merupakan anak kedua daripada 4 orang adik-beradik. Profesor Dr. Charles S. Vairappan memulakan pendidikan rendah di Sekolah Rendah Assumption dan Sekolah Menengah Assumption di Butterworth sebelum meneruskan pendidikan sekolah menengahnya di Sekolah St. Mark, Bagan Ajam. Selanjutnya, meneruskan pengajian tingkatan 6 di Sekolah Menengah Dato' On sebelum diterima melanjutkan pengajian Sarjana Muda Sains Gunaan dan pengajian Sarjana dalam bidang Biokimia Rumpai Laut di Universiti Malaysia Sabah, di Universiti Sains Malaysia, Pulau Pinang pada tahun 1995. Kecemerlangan akademik yang ditonjolkan oleh Profesor Dr. Charles Vairappan membolehkan beliau mendapat beasiswa untuk melanjutkan pengajian Dr. Falsafah dari Kerajaan Jepun di bawah program Mabusho Scholarship di Hokkaido University. Beliau juga telah memperolehi sijil dalam bahasa Jepun pada tahun 1998 sebelum memulakan penyelidikan peringkat Doktor Falsafah. Di atas kegigihan dan kekentalan jiwanya, beliau berjaya menyelesaikan program pengajian Doktor Falsafah dalam tempoh 3 tahun dan memperoleh ijazah Doktor Falsafah dalam bidang kimia produk semula jadi dari Graduate School of Environmental Earth Sciences, Faculty of Environmental Earth Sciences, Hokkaido University. Pada tahun 2006, beliau telah menyempurnakan kajian postdoctoral di bawah Japanese Society for Promotion of Science, JSPS Postdoctoral Fellowship selama 18 bulan dan telah menjalankan penyelidikan di Graduate School of Agriculture Science, University of Tokyo, Japan. Hadirin yang dihormati sekalian, perjalanan karier akademik beliau bermula di UMS pada 2 Januari tahun 2000 semasa tahun kedua pengajian Dr. Falsafah, iaitu sebagai tutor di Institut Penyelidikan Marine Borneo IPMB. Kemudian kerjayanya terus berkembang apabila beliau dilantik sebagai pensyarah universiti pada 2 April 2001 dan kemudian dinaikkan pangkat ke pensyarah kanan pada 18 Julai tahun 2005. Langkah ke dunia akademik beliau semakin menyerlah apabila dinaikkan pangkat ke jawatan Profesor Madya pada 9 Jun tahun 2008 dan jawatan Profesor penuh pada 6 September tahun 2013. Ketokohan yang ditonjolkan sepanjang menjawat jawatan akademik telah membolehkan beliau pada tahun 2011 dilantik oleh pengurusan universiti untuk menjawat jawatan timbalan pengarah sehingga tahun 2013 dan menyandang jawatan sebagai pengarah di Institut Biologi, Tropika dan Pemuliharaan bermula pada tahun 2013 sehingga tahun 2017. Hadirin yang dihormati sekalian, Profesor Dr. Charles juga telah menyilia lebih daripada 10 orang graduan Doktor Falsafah Sains dan 30 orang graduan Sarjana Sains. Ketokohan beliau turut diperlihatkan apabila dilantik dalam pelbagai jawatan kuasa, antaranya Majlis Biodiversity Sabah, ahli jawatan kuasa penasihat bioteknologi Sabah, Lembaga Pengarah Eksekutif Federasi Bioteknologi Asia AFOB mewakili Malaysia semenjak tahun 2016. Beliau juga merupakan ahli persatuan-persatuan berikut: Persatuan Kimia Amerika, Persatuan Fikologi Jepun, Persatuan Fikologi Gunaan Asia Pasifik, Persatuan Rumpai Laut Antarabangsa, Persatuan Ekologi Tropika Jepun, dan Persatuan Produk Asli Malaysia. Hadirin yang dihormati sekalian, Profesor Dr. Charles juga telah menerbitkan lebih daripada 130 artikel akademik mengenai penyelidikan beliau yang berwasit, bab dalam buku dan beberapa buah buku ilmiah, dan penyelidikan inovatifnya telah membuka jalan untuk pemindahan teknologi probiotik fortified marine silage ke syarikat aquakultur tempatan melalui geran dana tekno oleh Kementerian Pertanian Malaysia pada tahun 2013. Di samping itu beliau telah memenangi satu semi grand prize 10 pingat emas dan 3 pingat perak dalam pertandingan inovasi antarabangsa semenjak tahun 2004 sehingga tahun 2017. Beliau juga mempunyai 2 tanda dagang ataupun trademark untuk produk di kelas 16 dan kelas 31 dan 5 paten bagi inovasi dalam kimia produk semula jadi. Profesor Dr. Charles juga terlibat sebagai perunding bagi komuniti Pasifik Selatan, Pulau Solomon dan Digosa Texturan Sistem Kagil France untuk membantu dalam wabak penyakit rumpai laut di Filipina, Indonesia, Zanzibar dan Pulau Solomon. Beliau turut diundang sebagai plenary speaker dan keynote speaker dalam pelbagai program ilmiah antarabangsa, kebangsaan dan dialog kerjasama Selatan-Selatan di UNESCO Nairobi pada tahun 2014. Hadirin yang dihormati sekalian, kecemerlangan yang diperlihatkan oleh Dr. Charles sepanjang 20 tahun berkhidmat di UMS telah membolehkan beliau merangkul beberapa anugerah di pelbagai peringkat, antaranya anugerah tokoh penyelidik bagi kategori penyelidik 2010, anugerah tokoh penyelidik bagi kategori malim pada tahun 2013, anugerah khas perkhidmatan cemerlang pada tahun 2013 dan anugerah penyelia terbaik kategori sains pada tahun 2015. Beliau juga merupakan pengamal NLP bertauliah oleh NLP Academic of Malaysia dan juga merupakan profesor pelawat di The University of Tokyo dan Gaken University, Jepun. Selain itu, beliau juga memegang jawatan sebagai Ambassador of Hokkaido University dan Network Ambassador for Kagoshima University semenjak tahun 2017. Akademi Sains Malaysia juga telah mengiktiraf prestasi penyelidikan beliau dengan penghargaan dan mengangkat beliau sebagai salah seorang dari saintis penyelidikan terkemuka Malaysia 2020, Top Research Scientist of Malaysia 2020. Kerajaan Jepun juga telah menganugerahkan beliau Japanese Foreign Minister's Commendation Award 2021 bagi peranan beliau dalam kerjasama dua hala di bidang penyelidikan antara dua negara Malaysia dan Jepun. Atas segala sumbangan dan bakti yang telah dicurahkan kepada Universiti Malaysia Sabah dan masyarakat, pada hari yang bersejarah ini, Profesor Dr. Charles S. Vairappan telah diakui sebagai salah seorang sarjana daripada UMS yang cemerlang khususnya dalam bidang kepakaran beliau, iaitu kimia produk semula jadi. Tanpa melengahkan bicara, saya bagi pihak majlis dan juga bagi pihak yang berbahagia Datuk Naib Chancellor dengan segala hormatnya mempersilakan yang berusaha Profesor Dr. Charles S. Vairappan menyampaikan syarahan perdana beliau yang bertajuk Ecological Chemicals as Ecosystem Function Mediators and Potential Lead Pharmaceuticals. Dengan segala hormatnya dipersilakan. Sekian wabillahi taufik wal hidayah. Assalamualaikum warahmatullahi wabarakatuh.
Salam sejahtera dan selamat pagi. Yang berbahagia, Timbalan Naib Chancellor Akademik Profesor Dr. Rashid Mail, mewakili yang berbahagia Datuk Naib Chancellor Universiti Malaysia Sabah, yang berusaha Profesor Makdia Dr. Monica Sulaiman, Pengarah Institut Biologi Tropika dan Pemuliharaan, Div jemputan, rakan-rakan sekalian. Dengan izin Tuhan, kita telah dapat berkumpul pagi ini untuk menyempurnakan satu lagi aktiviti akademik yang bermakna untuk universiti dan diri saya, Syarahan Perdana ke-10. Pencapaian saya dalam bidang akademik dan penyelidikan selama 21 tahun di UMS telah disokong oleh ramai rakan saing dari pelbagai agensi, baik di Malaysia atau luar negara. Maka izinkan saya untuk meneruskan penghargaan saya dalam bahasa Inggeris agar dapat difahami oleh rakan-rakan yang mengikuti syarahan ini melalui media sosial dari luar negara. I am blessed to be surrounded by many extraordinary people in my life. Without them, it would not be possible for me to do what I do and to advance my research in my quest for new knowledge in natural product chemistry. For all who have helped in shaping my ideas, encouraged me to dream big dreams, and aided in realizing them, I am deeply grateful. I must offer special thanks to my research students, co-researchers, colleagues, and collaborators. My gratitude also goes to Professor Datuk Chemis Technologies Dr. Taufik Yap Yun Hin, Vice Chancellor of Universiti Malaysia Sabah, for his support and encouragement to hold this inaugural lecture. Various granting agencies have provided research funds to accomplish many interesting projects, and I'm indebted to them. Deep sense of gratitude is due to my teachers, Professor Dr. Minoru Suzuki, Hokkaido University, and Professor Dr. Shigeki Matsunaga, the University of Tokyo, for their kindness in sharing their wisdom in research, philosophy, and life. I'm also thankful for all the challenges that came my way, as these were important catalysts in my growth to look beyond any obstacle to progress in life as a researcher and person. It is my pleasure to record my appreciation to few friends who have been instrumental in my research advancement, particularly Professor Dr. Ridwan Abdul Rahman, who's here with us. He's the one who spotted my talent and brought me to University Malaysia Sabah way back in year 2000. Professor Dato Dr. Muhammad Ali bin Hassan from University Putra Malaysia, a dear friend, a mentor, and a teacher. My friends in Japan, Professor Dr. Tatsufumi Okino, Hokkaido University, Professor Dr. Toshiyuki Hamada, Kagoshima University, Professor Dr. Indapal Singh, NAIPA, Associate Professor Dr. Takashi Kamada, Shizuoka Institute of Science and Technology, and Associate Professor Dr. Takahiro Ishii, University of Ryukyu, Japan. I'm also pleased to take this opportunity to thank my high school biology teacher, Professor Dr. Fong Sun Fok, who's here, who has been with UMS for some time now, for his encouragement since my early days in Penang, not only as a teacher, but also as a friend and mentor. I would also like to thank Dr. Maklarin Lakim, Director of Sabah Park, Dr. Rahim Matsar, Chair of Academic Science Malaysia, Sabah Chapter, and Professor Dr. Rosna Ismail, or more fondly they call Kak Ros, our former Deputy Vice Chancellor of Research and Innovation, for attending this talk. I would also like to thank the scientific committee that facilitated the review process of this inaugural lecture book, especially Professor Dr. Rashid Mail, who chaired the committee, and Ms. Lindsay Loren, who assisted in the final edition of this book. Finally, I would like to acknowledge with immense gratitude the support and love of my family, my beloved wife, Anita Susan Christian, my inspiring sons, Isaac Ivan, who's not here, he's in Labuan, Gabriel Enoch, and Noah Eaton. For without their love and understanding, all this would not have been achieved or been meaningful for me. Finally, I'm immensely humbled as I glorify the grace and blessing of Almighty God. Thank you. So with that, I will go to my talk today. Title of my talk is Ecological Chemicals as Ecosystem Function Mediators and Potential Lead Pharmaceutical. I know it's a mouthful of jargons, but it will highlight the most important aspects of research that I've done in the last 21 years in UMS. So basically the talk consists of 5 parts. First, introduction of the ecosystem services and ecological processes. Second, the diversity of metabolites from terrestrial plants and animals. Third, marine metabolites. Fourth, the lead pharmaceutical that we can obtain from natural product chemistry. And fifth, the conclusion. Human well-being depends very much on nature and the benefits that it provides. However, the relationship between human biodiversity and its habitat hinges on a complex and dynamic balance. After over 2 decades of debate, there's now a broad consensus that biodiversity brings about positive effect on ecological function, stability, and resilience in ecosystem. Ecosystem services are the benefits that humans obtain directly or indirectly from various ecosystems, and it provides a foundation for improving our well-being and achieving sustainable development. Both terrestrial and marine ecosystem provide many critical products and services, as well as less visible services that are essential for human being. Ecosystem services are classified into 4 major categories: provisioning, supporting, regulating, and cultural services. But the complexity of their mutual relationship could only be fully visualized if we understand their synergy, their interdependence, and trade-off interactions within the ecological hierarchy. The availability of these ecosystem services depends closely on the functioning of biological capitals that defines the nature of ecosystem and various components of our biosphere. Proper management of these biological capitals is important in maintaining the health of the environment and human well-being as we progress into the future. Since ecosystems vary in their species composition, the services that ecosystems provide can also vary in nature, time, and space. Many more ecosystems are actively managed for specific purposes, but because of the interaction between species and ecological processes, optimal management of ecosystem for one service may have consequences for the delivery of services provided by other species. Lately, strong consensus has been achieved in accepting that ecosystem services are derived from biophysical structures, its organisms, and ecological processes that defines the mechanics of ecosystem function. It is evident that ecological processes are the very pulse that sets the rhythm of the ecosystem function and eventually the quality of ecosystem services that we receive. This slide shows that the island of Borneo is probably the third largest island and has got one of the best biodiversities in the world. If you look at the history, Borneo was connected to the Southeast Asian other islands through the Sunda Shelf during the Pleistocene period and became a corridor for human and biodiversity. As organisms travel, they have migrated into the island of Borneo, and in the middle Pleistocene, the connecting Sunda Shelf was a savanna grassland. Then it became a rainforest in the late Pleistocene era. And when there was melting of ice and increment in the sea level, the islands emerged. Borneo was separated from the other parts of Asia. So if you look at the island of Borneo today, you'll find that there's a lot of biodiversity, particularly along the Crocker Range corridor, and Sabah is known to be one of the hotspots for biodiversity. Now, we have about 15,000 species of flowering plants, 3,000 species of trees, 267 species of Diprotropika, and there are lots of endemic organisms, 44 endemic mammals, 37 species of birds that are endemic, and about 19 species of fish that's endemic. But the organisms that live in the tropical rainforest, they don't live in a harmonious environment. They interact. There's a lot of interaction between them. The interaction could be communication. It could be predatory. They might graze on plants. They could even compete for mating rights. And during this interaction, a lot of chemicals are being used. So I went on to study the chemicals that are being used by plants and animals in the tropical rainforest, and to have a better understanding of the mechanics of how the chemicals work. Having said that, the marine biodiversity is also very unique. As I've mentioned, the Sunda Shelf, when the islands emerged due to the rise of the seawater, the former rainforest covered Sunda Shelf now became submerged in water, and eventually it became coral reefs. And we have one of the most productive coral reefs in this region, particularly in Sabah, as can be seen in the slide, the map of Sulu Sulawesi marine ecoregion. It's one of the most productive and rich with a high diversity of species. Knowledge of species, its unique distribution, their resources, and it sustains a huge value in economical terms. That's why the region is now called Sulu Sulawesi Marine Ecoregion. It's no more Sulu Sulawesi Coral Triangle. And similarly, the marine environment is also a very dynamic environment where organisms interact with each other. My early days in IPMB, I took these photographs in 2002, and these photographs were taken just outside the IPMB jetty that is in Pulau Sepangga. As you can see, I was very fascinated with the interaction at the coral reefs, where you find sponges, soft corals, hydras, and many more invertebrates and plants living together. As you can see the slides below, there was interaction between barrel sponge and brittle star. Now, barrel sponge are known to produce toxic compounds, but brittle star could actually stay in them and they could survive. In fact, they were using some of these compounds as their defense tools. Similarly, nudibranch and also the squid, in their mating dance, not only force is used, but also chemicals are used for communication. And of course, Aplysia. Aplysias are known to graze on Laurentia seaweed, and they use these chemicals as their defense tools. And finally, the Christmas tree worm. When you try to touch them, they will retrieve because they can sense the amino acid that's at your fingertips. So this was the beauty of chemical ecology that I came to know while I was at IPMB. Later, I also found that soft corals are also very interesting. They're very soft but not eaten by any fish. And they are also very potent with a lot of biological activities. And eventually, I started working on them and I found a whole diversity of secondary metabolites that are very potent and could be used as lead pharmaceutical. That will be the later part of my talk. And we also have seaweeds, example, the green algae, the grape of sea, and so on. And I also worked on Polycarpa. Polycarpa is a tunicate that resembles a beating heart. The purple ink that you can see is very dynamic and it moves when it inhales the water, and it's seen like a heart that is beating in the sea. All this fascinated me, and it gave me a time to think about what I wanted to do, and I decided to work on chemical ecology that leads to drug discovery. So today I will going to share with you some of my findings in this area. So the research aim of my work in UMS was deliberate on the advances achieved in understanding the role on chemicals in plant-animal interaction. Today I will do that. Second, to describe the contribution of these ecochemicals to the ecosystem function in a given ecosystem. This is the aim. Today I will try to deliberate on this. Then the questions that I try to ask or to answer are these ecochemicals synthesized for a specific or broad range of functions in the ecological processes, and is there an applicable link, that is there a possibility that we could actually utilize some of these compounds. With that, let me move on to first put the basic understanding. What are the ecological chemicals? Now, ecological chemicals could be divided into two broad groups. One are the primary metabolites. Primary metabolites are organic molecules of natural product that are directly involved in normal growth, development, and reproduction. These compounds could be, as shown here, vitamins, lactic acid, acetic acid, and other organic acids that are produced by the organism and used for various biochemical processes. But the secondary metabolites are specialized organic chemical, meaning to say the organism has to invest lots of energy in producing these compounds. And these compounds are basically used as tools to defend themselves, to communicate, to conduct quorum sensing, and for reproduction. They're very important chemicals. With that, let me move on to the ecological chemicals in terrestrial ecosystem. As I've mentioned, Borneo is a very rich land with high density of biodiversity. So my study took me to places and to different ecosystems. First, primary forests, logged forests, secondary forests, Kerangas forest types, peatland forests, mangrove forests, and montane forests. So I'll touch some of these aspects. And in the primary, logged and secondary forests, I studied the importance of dung beetles as ecosystem service providers. In Karangas forest, peatland forest, I discovered some very important herbs and gingers and plants that could be used as secondary metabolites for human application and also for chemotaxonomical utilization in biosystematics of these plants. In mangrove forests, I found that the mangrove leaves has some steroid compounds that could elevate the blood pressures in proboscis monkey, particularly the male ones. And in montane forests, I studied mosses and lichens. To start off, essential oil in herbs. Now, all of us would have heard about essential oils. We have used them. And these are very important chemicals produced by plants, particularly herbs. And in my laboratory, I've studied, analyzed close to about 400 different types of plants, and we've got a variety of essential oils from these plants. These compounds or these oils have got very diverse application. They could be used as antifungal, antimicrobial, anti-cancer, and also as wound healing compound, and also in cosmetic application. Now, some of these major compounds have been used in Malay traditional medicinal practices. Here you can see the four main herbs that are used in Malay traditional medicine are the halia bara, and at times it's also substituted by the normal ginger, gerangau, Acorus calamus, or bonglai, Zingiber casumina, and chikor, Kaempferia galanga. These are the basic herbs used by a Malay medicine man, and there's not much literature written on these herbs, so I wanted to explore this. Now, halia bara has a lot of secondary metabolites and essential oil, particularly Alpinia purpurata has compounds resembling pepper compounds or alkaloids, pepperin type of compounds, and Zingiber officinale has got lots of chargol and gingerol. These are phenolic type compounds that are very pungent and has got very important pharmaceutical properties. Gerangau, bonglai, and chikor has got lots of essential oil, and it justifies why the Malay community has been using these plants as medicinal plants for a long time. And our analysis showed that Acorus calamus has cis asaron, the compound, number 3 compound, to the level of 90% of its essential oil, almost pure. This is unthinkable in any natural plant material. We were very surprised. In fact, Casumuna, a bonglai, it had sabinin 30%. 30% is very high. And also terpinin, also 30%. Galanga, a chikor, Kaempferia galanga, that is called chikor locally, had cinnamic acid. But 60% of cinnamic acid is unthinkable. It is really very potent. And of course, halia bara had citral and nerol in its essential oil. And all these could be used as important essential oils. Herbal and lower plants. Besides these 4 main herbs that the Malay community has been using, there are also a lot of other herbs that have been used for various applications. So I ventured into different herbs. This is some work done on curcuma. Now, as we know, curcuma has been known to produce lots of curcumin. Yeah, curcumin, the structure shown there, curcumins are actually diaryl heptanoids, meaning it has got 2 benzene ring connected by 7 carbons. And there's a lot of functional differences in the structure that produces a whole diversity of curcumins. So when we eat curcumin or when we eat turmeric, we're actually ingesting a variety of metabolites in the group of curcumin. So it has got lots of potential. But if you were to look for any literature in Malaysia or in Southeast Asia, you'll be surprised. You will never get one document that has got sufficient information for all the essential oils. So what we decided to do is to look at the 8 most important curcumas in the Asian countries. So these were Cucuma rotunda, it's also known as temu kunci, Cucuma longa, which is our normal turmeric, Cucuma aeruginosa, which is also called as Durga manjal in India, Cucuma xanthorrhiza, Cucuma heniana, Cucuma cassia, Cucuma zeodora, and Cucuma manga. And that particular last one, Cucuma manga, really tastes like mango. So I did I extracted the essential oil, and we used the latest instruments that we have in our laboratory to analyze the essential oil composition. And we introduced a new analysis where we looked at we performed a statistical analysis to make a heat map of all the essential oils. The axis on the extreme right shows the variety of cucumas that were analyzed, and as you can see, the dark spots and also the light dark spot shows the intensity of various compounds that are present. And we were able to identify that most of these compounds in this genus could be identified using one set of compounds, so they belong to a particular genus. So there's a chemical marker that represents this genus, but there were also some other chemicals that you can see that are dark blue in that particular diagram that represents other features that could differentiate the other members of the cucuma, the 8 cucumas that we have studied. So this was a very important tool, developing a heat map and analyzing them. We were able to use essential oil to show some of these oils could be used as chemotaxonomical markers, while others could be used to differentiate different species of cucuma within that genus. This is something new. We'll be publishing this very soon. Then we went on to do NMDS analysis. This is a statistical analysis. We were able to conclusively show that there were 4 clusters based on the chemical. Cluster 1 represented by Cucuma aeruginosa, Cucuma cassia, and zeodora. Cluster 2 by longa and heniana. Cluster 3 was manga, and cluster 4 rotunda and xanthorrhiza. And the figure on the right shows lots of lines, I know. Yeah, the blue lines represents a certain group of compounds, maybe hydrocarbons. Some represented alcohol, and based on the intensity of the lines, we were able to identify which were the major compounds in some of these species. These are all latest information that has not been published, and these are very relevant to the science of taxonomy. So we have introduced a new science called chemotaxonomy, and it can be used to differentiate between genus and within genus. Next, let me talk about something that we all know, the curry leaf. Curry leaf is very often used for its aroma when we cook. The essential oils are very important. So it's basically carophyllene and humulene. When we cook something, the aroma is from this essential oil. But they also have got a very high concentration of a compound called alkaloids. Alkaloids are very important. Yeah, here we found almost about 6 to 8 alkaloids, and 3 were new stereochemically. And here I would like to talk about introduce one particular compound that is called mahanimbicin. This was very unique. It had a very interesting chemical profile, and we were able to use this compound in mouse model to treat inflammation. So we wounded the mouse and we applied this compound in a very low concentration, and it was able to assist the mouse to recover recover its wound. So it had very good healing potential, and it also was very important for the recreation of that skin surface. Therefore, besides eating curry leaf as part of the cooking process or just for its aroma, it is also a good idea to consume the leaf as a chutney or something like that, so that you'll be able to benefit from the mahanimbicin and alkaloids that are present in this curry leaf. Thank you. Next, our lengkuas, Alpinia galanga. Well, this particular aspect, I did not want to work on this. I didn't want to work. But one of my final year students insisted that she wanted to work. Then with heavy heart, I said, yeah, sure, why not? We're not going to find anything new. So, but to our surprise, when we started working on it, the first 3 compounds that you see are all very new compounds. Has not been reported in science till today. We were very surprised. Even a very common ginger or a very common alpinia, which is the lengkuas in this case, can also produce new novel chemicals. So there's new compounds everywhere, only if we look into them. And these compounds, the 3 new compounds that I've isolated and identified, were shown to have very good anti-inflammatory potential. I will talk about anti-inflammation in the 4th part of my talk. Next, the mountain forest. Here I work with Dr. Monica on mosses. One of my PhD students we co-supervised one PhD student. They worked on liverwort. And liverwort is a group of very interesting plants. They are primitive and they are believed to have originated from the marine environment. And I just want you to remember, when you look at the slides, the pictures on the right hand side shows microscopic pictures of the surface cellular arrangement of this particular liverwort, and these are all granular oil vesicles. And later I will show you that seaweed has also got this. So I was very fascinated with the oil granules, so went on to study the chemistry of this liverwort. Monica helped for the identification of it. So we've got 5 here. First, Mastigophora. This particular liverwort, when we analyzed and studied them, they produce 3 different chemical skeletons. Now, it looks simple, but it's not easy to isolate them in pure form and to illustrate the structure. We were fortunate that we have got the tools in UMS. We have 600 megahertz NMR. We have high resolution MS and variety of other instruments. We were able to do this in a relatively short period of time. So Mastigophora had about 8 compounds. Now the herbitin compounds are mostly also called as copperan, and these are found in lots of marine plants too. And this group of Mastigophora are also known to produce chlorinated compounds, which are very, very rare. Next was Chandanotus, and this is another very interesting group. We can only find sembrane in the marine organism, particularly soft coral. But we were fortunate that we could find Chandanotus, which is a liverwort in Kinabalu, that produced sembrane molecules. It's quite rare. Next, Gotchiella. If you go to Gunung Alap, the whole slope in Gunung Alap is full of Gotchiella. It's a red meadow, beautiful. And we were fortunate to have isolated 6 claridane skeletons from this Gotchiella, and we have published a couple of papers from this. And these are very potent compounds with biological potential. Very interesting structures. Next, Cystochyla. Cystochyla are being used in certain part of the world as part of community traditional medicine. And these two had scleridane, secoscleridane, and dolabellin. I think a marine biologist will know dolabella. Dolabella is actually a sea hare, and dolabellin originated from a sea hare. So here we see a compound that has got marine origin could also be found in liverwort, which is in the highlands. So we can show the connection between marine organism and highland liverwort. Acrobolus, and these had very interesting 6-membrane compounds, very bioactive, and we were able to isolate some and we have published. And these are very chemical structurally is very important and very interesting with the stereochemistry. So they also showed a lot of biological potentials. Now, to put them all together, in total, we studied about 6 to 7 or almost 8 different liverwort. Here I show some of them with their signature skeletons. So skeletons are bigger group chemicals that we can identify them based on the chemical properties. And during our field work, we found that some of these leopards were grazed by slugs. Some of these compounds are used by the slugs for their own defense, and some of these compounds also repel some insects and other organisms from feeding them. And some of these compounds which has got double bond could also be used as absorbers of UV light. Next, lichen chemistry. Lichens are actually a symbiotic organism. We always say the lichens keep microalgae as their prisoners, because the lichen is actually a fungal which is called a microbiont, and they keep the microalgae within them as prisoners, where the phytobiont will photosynthesize and produce nutrients, and the fungus will utilize the nutrient from this photobiont. So we were very interested in this because about 6-7 years ago, a researcher from Natural History Museum London came, Dr. Holger, and he introduced us to this particular unique plant. So I've studied two broad genus of lichens in Sabah, and this is on Styrocaulon. Styrocaulons are also known as snow lichens. They look like snow. Very nice, very beautiful Styrocaulons. And here we were fascinated to find that in Gunung Alap there were two different species of Styrocaulon, and they were growing side by side in abundance, and they had totally a different chemical profile altogether. Here you can see these are the Styrocaulons that were collected from Gunung Alap. One had the ability to absorb UV light, very, very obvious ability. The other one could not absorb any UV light at all. And the compounds that absorb UV light, you were able to identify these compounds, and this compound has got lots of cosmetic value, could be used as UV absorbing compounds for cosmetic production. And these are found in Gunung Alap. Beautiful organism. Next, I'm going to talk about something that is very interesting. Probably not before lunch, but anyway. So this is on homing signals for dung beetle. All right, dung beetle, yes. All right. So as you know, Sabah is a hotspot for biodiversity. Tropical rainforests are beautiful, but we should also realize that it's being replaced gradually. So we have created a landscape gradient where you have primary forests, logged forests, secondary forests, degraded forests, and finally plantation. So the organisms that live in this landscape gradient also changes from high biodiversity organisms to a more monotonous or less diversified organism as we move along towards the oil palm plantation. Now, I was interested to look at dung beetles because dung beetles are important ecosystem service providers. They work on the dungs from animals, they remineralize them, and then they incorporate them in soil. In that way, they return the nutrient back to the forest. Very important ecosystem service providers. Yeah, as I mentioned earlier, these are some of the services supporting or regulating services which are very important. And we wanted to know what was the diversity of dung beetle in primary forest, that is the Malayau Basin plot, Belian plot. We had 470 individuals with 21 species, very good. Even Kalabakan fragmented forest, we had about 110 less individuals, but the species composition was relatively good. But you can see the oil palm plantation, where we have less species and less individual, which is to be expected because oil palm plantation, you don't have many animals. But what is important is for us to understand how these dung beetles work. And for our surprise, when we did this study, we used human feces as the bait. So when we use them, we bait them, within seconds you can find buzzing sound of dung beetle coming. But when we use elephant dung, it can take the whole 1 day or 2 days, you could hardly find any dung beetle. You know, if you're lucky, maybe after 2 days you could find some dung beetles. So I was very intrigued. I thought there must be something in the air. It has to be something about the volatile compounds. So from then on, we thought about how are we going to go about this, and we devised some experiments. And I worked with some very interesting people from various parts of the world. Dr. Eleanor from Oxford, and Professor Sakai Kenji from Kyushu University. He's a microbiologist. He helped us to identify the bacteria. And Eleanor is an ecologist and also a dung beetle specialist. She assisted in the identification of dung beetles. So we studied about 10 different dung types. Most of the dungs were collected from the zoo with the wildlife department of Sabah. The rhino dungs were collected from Bora, and the bear dungs were collected from Bear Sanctuary in Sepilok, and we baited them, as you can see some of the slides where we go down to places like Malayao Basin and Kalabakan. We baited them, and we found out that some of these dungs had different profile compared to the others. So this is how we analyze them. We use sophisticated instrument called GCMS, and we use a very interesting technique, gas-tight vials where we put in and robotically it is analyzed. And some of these VOCs from dungs are very difficult to analyze, and we had to do some twitching and buying of certain columns. We were able to identify most of the VOCs. Now, dung beetles could be categorized into 3 different categories: dwellers, tunnelers, and rollers. So they behave differently. They also are attracted to different types of scent. So here we show the attractiveness of the dung. As you can see on the graph that is on the right, you can see the top first 3 lines are the primates: human, orangutan, and I think it's human, orangutan, and it's a rhino. They were most attractive. Dung beetles immediately go to them. But we look at the sun bear, it is less attractive. So there was a whole diversity of attractiveness in the dung. So we wanted to relate this to the compounds that we analyzed in the GCMS analysis. And we did a homing network of dung. The bars that you see on the left-hand side graph, the yellow ones are for herbivores, the pink ones are for omnivores, and there was 1 carnivore. And we were able to connect this to the network preference. The bars, the black bars on top are actually the dung beetles. I could not put all the names, so I put numbers. So you can see the thicker the line, these are preferred dungs by the dung beetles. We were able to connect, make networks, and we were also able to connect the dung type to the functional group, meaning the aroma or the smell or the fragrance in dung that attracts the dung beetle. For the dung beetles, these are fragrance. For us, these are not so important. So we were able to connect them to show that some of these VOCs are being used as homing chemicals. Now, we also tried to do some correlation activities, and what we found out that most of the dung beetles are generalists. They will just eat any dung. But there were also some very specific dung beetles. Like here, we can see one that is very close to the proboscis monkey, Copris. Copris is a nocturnal tunneler dung beetle, and it showed very high correlation with proboscis monkey. And Prof. Henry could definitely confirm this, that proboscis monkey, when at night, they don't move much. They nest in certain part of the, by the riverside probably, and they tend to drop lots of their dungs. So that's the ideal time for dung beetles, which are nocturnal, to come and feed on them. So it makes perfect sense. So we were able to know that proboscis monkey has got releases a lot of hydrocarbon and also carboxylic acid in their dung, and that attracts this particular species of dung beetle. So with such a correlation, we could also explain the other 4 dung beetles that showed correlation to certain dung type. So in essence, we were able to justify or give scientific reason as to why dung beetles prefer certain types of dungs. Of course, there are lots of generalists, but nevertheless, there were some specific ones that go for specific dung types. And we also correlated this with the bacteria. Professor Sakai helped us to do next generation sequencing of bacteria, and we were able to map the bacteria in human feces, sun bear, and some of these primates to justify that when you actually hang a dung in the field, the kind of bacteria that act on them and releases the chemicals. We were able to do that. Next, I would like to go to the marine ecological chemicals. This is very interesting for me. It's very close to my heart. And I've been working on Laurencia and relationship between Laurencia and sea hare, cultivation of seaweed that Prof. Ridwan started. And some of these had shown very wonderful results. Now, seaweed diversity in tropical Borneo is very interesting. Malaysia has got 4,675 kilometers of coastline. And in Borneo, we have about 2,607 kilometers of coastline. And as I've told you earlier, the coral reefs are beautiful, and we have various ecosystem types. And we have coral reefs, we have shallow seascapes, we have mangrove forests, and degraded coral reefs, which are ideal for growth of algae. So in total, we have about 456 taxa of seaweed belonging to 72 families. Cyanophyta are actually blue-green algae. They are filamentous algae, 12 families. Chlorophyta, green algae, 16 families. Ochrophyta, formerly known as Phaeophyta, brown algae. It's called something different now. So 8 families, and Rhodophyta, 36 families. Now, the commercially important seaweeds are the red algae. As you can see here on the extreme right are Gracilaria, and you can see Capophycus are being cultivated. And also you can also find some green algae which are being cultivated, like Kaulepa, which is also known as Umibudo in Japan. So to start off with, the first research that I did in UMS was to work on Laurencia secondary metabolites. Laurencia are red algae. They are known to be potent producers of secondary metabolites. They've got this enzyme called bromoperoxidase enzyme, and they produce these beautiful halogenated compounds. And these compounds, as I mentioned earlier, when I spoke about liverwort, this group of seaweeds also synthesize and keep their compounds in that vesicles that you can see on the right hand side, a microscopic picture. And they keep all these compounds in beautiful sacs, and these are arranged on the top of the cellular layer. And when an organism, insect, or bacteria or fungi touch on them, they break, and then the chemicals will react with this organism and will kill the organism. This is a defense compound of red algae. And we did chemical profiling. There are about 4 major species of Laurencia: Snecki, Majuscula, Nangi, and Similis. They all showed diversity of secondary metabolites, and I will show you quickly the skeletons that I found in some of these compounds. Now, the Laurencia snecki produce basically sesquiterpene halogenated with bromine, and these are called syndrane skeleton. And there were some other sesquiterpenes which were not syndrane, but they had functional groups like hydroxyl and also halogen. This is Laurencia majuscula, totally a different group of compounds. These are all chemigrane, halogenated, brominated, and chlorinated with various group of functionality. Very important, very potent compounds. This is Laurencia nangi, greenish color, but they are red algae, not green algae. They produce totally a different mixture of compounds. The compound that you see on the left is called estogenin. The one right top is called bromoaran, bottom diterpenes. And they also produce sesquiterpenes, triterpenes, and also other rearranged metabolites. And the last group of seaweed called Laurencia similis. And this particular group of seaweeds are similar to the one that is found in Okinawa, which is called Sozonohana, and they produce brominated alkaloids. These are the only seaweed that produces alkaloids. And in Sabah, we could only find 3 alkaloids with brominated alkaloids. Now, we did the same thing. We did various analyses. We were able to show the distribution of seaweed species, sampling location, number of compound, and number of structure. In essence, we wanted to use these chemicals to show that this could be used as chemotaxonomical markers, and we were able to do that. We also mapped them using a heat map and cluster analysis. We were able to show that as far as Lawrencia snecki and majuscula were concerned, there was some variation. Similis was totally homogeneous, and we found that Nangi and majuscula showed overlapping in certain chemical skeletons. We did generalized linear model, mixed model, to show the effect of number of compound, and we found that effect of location was important, effect of species was also important. Then we did similar generalized linear model effect for structure, and we found that effect of location was not important in producing the kind of structures, but the effect of species was important. So statistical analysis was used as a tool to justify and to confirm certain pattern that we found in the chemistry. And this is not usually done. We wanted to use what is statistical tools that are available for biological study in chemistry, and to use it as a bridge to connect two different disciplines of science. And we also did some cluster analysis, and you can easily see that some of these compounds, particularly the figure on the right, you can see the green line in between the pinkish color. So it shows that there is some overlap between Lawrencia nangi and Lawrencia majuscula. So we confirmed that using some logistic regression model, and we were able to say that in terms of number of compounds, majuscula and nangi are different compared to similis and snecki, conclusively. And then in terms of number of structure type, Lawrencia majuscula and nangi are different compared to similis and snecki. So these are very important findings that we found from our study to say that halogenetic metabolites could be used as chemotaxonomical markers. Now, Lawrencia are also eaten by sea hares. You can see the photograph there. I think Prof. Ridwan will recognize that's Bujang there with Pushpa. And we collected some sea hare. And the moment you collect, you bring them to the boat, they will start to release a kind of a purplish ink, and the compounds are in the ink. So we were very fascinated with this. We brought them to the lab. And the second photograph shows that dissection of this organism in the laboratory, and then we did a quick extraction and HPLC analysis. We compared it with the seaweed, and as you can see, the arrows that are being showed, there were some similar compounds between the seaweed and the Aplysia. This is Aplysia dactylomela. These are gastropods. So what the gastropods do, they eat Lawrencia, they keep the compounds in their guts, and then they use them as their defense mechanism. They escape from fish, they escape from squid by releasing the purplish ink, which has got a very high concentration of halogenetic metabolites, to confuse their predators and to get away. And we were able to show chemically, we isolated the structures, we elucidated the structures to show that Aplysia has also got this ability to actually produce rearranged compound in their guts. So the compounds that we found from sea hare gut some were totally new, meaning to say these compounds were rearranged or chemically sort of say synthesized in their guts. It's a very interesting concept, and the sea hares were capable of doing that. This is where Prof. Ridwan comes in. The first photograph shows younger Ridwan, right? So when I joined UMS in year 2000, okay, I could not see it here. Okay, all right. So when I joined UMS in year 2000, Prof. Ridwan has already started working on a pilot project in Kudat, Bangi, with Prof. Ranjit. You can see this second slide. You can see Ranjit there carrying two lumps of seaweed, and these are commercially important seaweed, Capophycus alvarezi and Euchima denticulata. One is called cotonai, the other one is called spinosum, and it's being cultivated as a monoculture seaweed and in long lines, 100, 200 meters. And Prof. Ridwan was helping the community there, and I was involved in certain aspects, not really in the cultivation, but on the cosmetic production and chemical analysis of these. Now, this particular seaweed, Capophycus, are known to produce carrageenan. Carrageenan are polysaccharides. They've got a lot of commercial application. When you use your cosmetics, the gelling or the consistency is produced by the carrageenan. When you drink yogurt drinks or you use yogurt or any other dairy product, it has got lots of carrageenan in them. Then there are two varieties of seaweed. Capophycus, the one on top, produces kappa carrageenan. That's why it's called kappa ficus. And then the lower one produces iota carrageenan, which is the spinosome type. Spinosome has got less value because of the gel type, but kappa ficus, which produces kappa carrageenan, has got lots of value. And Malaysia is ranked at number 4 major producer of kappa ficus after Philippines, Indonesia, and now I think Zanzibar has taken over. I'm not very sure about that, but the top 2 are Philippines and Indonesia, and probably Tanzania, Zanzibar, then Malaysia. Now, there are some major issues in the cultivation of seaweed: eye-size disease, epiphyte infection, and post-harvest handling. All this compromises the market price for seaweed. So, I'm a chemist. I did not want to work on epiphyte or seaweed disease, but I can still remember one fine day, Ranjit and Prof. Ridwan, I think it was a Monday morning, he came knocking at my door, and both of them can be very persuasive. They somehow asked me to have a look at a very grossly looking seaweed. You can see the photograph on the left, the D figure. My first look at the seaweed, that's how it looked like. Very hairy looking seaweed. I was not interested. I told him, look, I'm a chemist, I don't want to work on this. He said, no, just have a look. So I had a look at it and I just started playing around with it. I was able to dislodge the epiphytes from that particular seaweed. I was able to take some photographs. And after taking the photographs, what I did, I just emailed my friends in Hokkaido University. And to my surprise, the next day, all the species were identified. Professor Michio Mazda from Hokkaido University identified the epiphytes on the seaweed. And then I went on, because I was hooked up to this, so I went on to do some scanning electron microscope images. As you can see how the epiphyte goes into the kappa ficus. These are filamentous red algae. They are algae actually, filamentous, and they bore hole and then they go inside. And we were able to identify them as Neosiphonia savatieri. Yeah? And although the symptom of epiphyte infection was identified long time ago by Largo, by Doty, but they were not able to identify the species. So I was fortunate to stumble on this because of Prof. Ridwan, and I worked on it, and we were able to get some very beautiful pictures and identify the organism, and we were also able to document the symptoms, mode of infection, details of epiphytes, and also the abiotic factors that caused the outbreak of epiphyte. So what I found was that there's 2 episodes of explosion, epiphyte explosion. First is April to May, where the density is the highest, 60 epiphytes per cm square, and then October to November, about 25 epiphytes per cm square. So we were able to relate these to the drastic increment in abiotic factors or water salinity, temperature, or drastic drop. Whether it increases or it drops, it still affects the immune system. So therefore, the epiphytes which are outside the seaweed start attacking the seaweed because the seaweed's immune systems are down. And then we went on to see the source of it. Now, you can find this seaweed, this epiphyte infection in Sampurna, of course in Kudat. But you can never find this in Nunukan seaweed farms. Because in Nunukan seaweed farms, when I visited the farm, they did not have beautiful coral reefs around the farming area. The seawater was a bit cloudy and the coral reefs could not survive. So the source of the epiphyte is in the coral reefs. So could not migrate and affect the seaweeds. But in Sampurna and in Kudat, the coral reefs are beautiful, and this harbors all these epiphytes, and they were just waiting to attack the seaweeds when their guts were down. So the abiotic factors were the cause of their loss in defense mechanism. Then in 2005, eye-size disease hit the seaweed industry in Sampurna. So most of the seedlings were gone. So our farmers, what they did, they took the boat, they went to Indonesia and Philippines, they brought some seedlings from there, without quarantine, and that brought in a new strain of epiphyte, Neosiphonia apiculata. As you can see from the scanning electron microscope, the symptoms were really different, right? This is what some people call goosebump symptoms, and it really affects the seaweed. Drastic changes in the physiology of the seaweed, and the loss of biomass was very significant. So in 2005, new strain of epiphyte was introduced into Sabah waters. And in 2004, I went for a conference in Bergen. That's where I met Alan Critchley, that's soya flour, and Annie Hurtado. Alan was working for Degusa Texturan. Flour is from Zanzibar, and Annie is from Philippines. So they took me for a drink, and then we had discussion, and finally Alan said, okay Charles, we will sponsor you, we will give you a contract from Degusa, why don't you work on this? And Flour and Annie, they put their connections to networking together, and they were able to get for me seaweeds from Philippines, Indonesia, Tanzania, that is from Zanzibar, and they sent it over to my lab. So I was able to look at the symptoms and the type of epiphyte at my lab from the resources that I got from this team. So with that, I was able to show that the epiphyte that's in Philippines, Indonesia, Tanzania, and Malaysia were the same. Neosiphonia apiculata. And this particular epiphyte also caused secondary infection. So the moment it falls off from the seaweed, it leaves a hole, and then from that hole, bacteria sets in, and it destroys the whole seaweed. So we were able to deduce the kind of bacteria that destroys them and how it goes about destroying the thallus. So at the end of the day, we found out that the epiphyte infection facilitated in the drop of the carrageenan, the quality of the seaweed. So the infected seaweed produced less carrageenan, less viscous, and less value, of course. The farmers were totally in trouble. So how does this happen? So what happens is that the epiphyte penetrates into the cell, it releases the enzyme, and it cuts the carrageenan. Carrageenan is usually exists in 800 kilodalton, meaning the molecular size is huge. But when the epiphyte goes in, it pumps in enzyme and acid, it breaks the carrageenan into smaller molecule. 80 kilodalton, this is 10% of the original size, and also 25 kilodalton. With that, the carrageenan becomes so small, it loses its viscosity and its ability to gel. So there's no market value for that sort of carrageenan. So this was the problem. So we were able to establish how epiphyte reduces the quality of carrageenan in the infected seaweed. So how are we going to solve this problem, right? So these are some of the photos that I managed to find from the work that I did with my students. What we did, we thought there has to be an answer from nature. So we did a 2 years monitoring of the seaweed lines, particularly in Sempurna and in Kudat. So we wanted to learn from nature. So what we did, we meticulously studied the biofoulas. Biofoulas are needless seaweed that grows on the line. So usually the farmers will remove them. So we studied the seasonality, we looked at the abiotic factor, we looked at the presence and absence of pathogens, and also the chemicals in the water. Now, Carboficus, when it's cultivated in a high density, it also produces hydrogen peroxide. I'm not sure if Prof. Ridwan is aware of this. It produces hydrogen peroxide with stress, and that hydrogen peroxide is actually, it dissolves in the water, and it has got certain amount of impact on the coral reefs just immediately under the seaweed lines. So we were able to note some of these findings, and when we studied them, we found that certain biofoulas were there even when there was a epiphyte outbreak. So we were very fascinated with that. And we found that Lawrencea majuscula was there even when there was a huge heat of epiphytes. And we were surprised. We wanted to know why. So we studied 6 farms: Lohok Butun, Selakan Island, Boom Boom Island, and what do you call that place? Carrington Reef, Balabangan. Yeah, these are some of the places that we took the seaweed from, and we studied them. Terutuk. These are some of the areas. And we were able to identify the compounds as 4 halogenated sesquiterpene. The compound 1 and 2 are simple sesquiterpene with halogenation, and the 3 and 4 are vinyl bromide compounds. And we were also able to trace their concentration, and we were able to prove in the laboratory that these compounds could actually destroy these epiphytes, could be used as an alternative mode of control of these epiphytes. It's called cough spatialate. We did these experiments in the lab to prove the concentration and the type of compound that's needed as a defense in the seaweed cultivation line. Then we suggested to the fishery department and relevant agencies and also the International Seaweed Association that when they culture Copperficus, it is not actually a good idea to remove all the biofouls. Maybe most of them, but not Lawrencea majuscula. Next, I'll move on to soft coral. Soft corals are marine invertebrates, right? Cnidarians. These are found in large numbers in coral reefs of Sabah, particularly the Sulu Sulawesi ecoregion. And we were able to collect some of these and to look at their secondary metabolites. As I've mentioned, these are very soft invertebrates, but not eaten by fish. Sometimes some turtles eat them, but fish usually don't touch them. So what we did, we collected various types of soft corals and we isolated the compounds. We were able to categorize the compound based on their structure. And with that, with chemical profiling, we were able to show that some of these soft corals could be, compounds from soft corals could be categorized into terpenes, non-terpenes, sterols, and so on. Just to show some of the examples. Soft corals like Carbonella produce a lot of sesquiterpene, they call this Carbonella-based compounds, while Lamnalia produce lamnarin, and there was a lot of compounds from Lamnalia, and we were able to publish very interesting papers of the structure diversity, while Lobophyton produced a lot of sembrane-based. This is the compound that I showed earlier in liverwort that was there. It is still the same structure. It's called sembrane structure, and we found lots of it in Lobophyton. Then we have Sacrophyton, Sinularia, and Naftia. All these produced a mixture of sembrane, sesquiterpenes, and diterpenes. So we had, at the end of the study, we had lots of secondary metabolites from soft corals. And we were able to do the same thing we did with Lawrencea compound. We introduced them into a chemotaxonomical analysis, and we were able to show number of compounds, number of skeletons, versus the locations where they were taken. And we also did phylogeny analysis to show their phylogenical relationship together with the chemotaxonomical markers. So this is a new science. We were able to use chemicals, secondary metabolites as chemotaxonomical markers for biosystematics of soft coral. This is something new that we have introduced for the biosystematics of Lawrencea and soft coral. This is the carnation coral. It looks beautiful, very bright, very interesting, but you can't touch them. They're very thorny. We were able to identify some very potent sterols. There are a huge pool of sterols in this particular soft corals, in these carnation corals. They are found in abundance just around Sepangga Island. And we were able to identify them, and some of these sterols could be used as important lead pharmaceutical. This is just an example of two sponges that we studied. One, we were able to find some very interesting compounds which had both sterol property and also dissolving property in most aqueous solution, very toxic compound. And aptose produced aptamine, which is a very commonly known neurotoxin. So all these are found in Sabah waters. Next, the fourth part of my talk, lead pharmaceuticals. So now we know we have a lot of lifestyle diseases. We have issues with corona now, and we also have issues with inflammation, cancer. So we wanted to find novel compounds. We wanted to find compounds that are of importance. So as you know, as you can look at the table there, even compounds from Euchima, Iota carrageenan, are being used for antiviral. And there are a lot of other compounds from like yondeline and other compounds that are isolated from marine organisms that are being used as drug, and they are in various phases. Phase 3, some have got FDA approval, and so on and so forth. So marine marine organisms are known to produce a huge wide diversity of potential compounds. So we wanted to see the 2,000 compounds that we have isolated, whether this could be used for our human consumption. First, I'll talk briefly about anti-inflammation. What is inflammation? When you slam the car door on your finger gets inflamed, meaning the tissue is destroyed. There's a lot of force, so the blood is rushed into the place to repair that wound. So when that happens, you find some blue-black marks. If it's something minor, it goes off. But something major, then you need to look into it, put some medicine. But if it's left, if a chronic inflammation is left untreated, let it be a sore throat, let it be an ulcer in the stomach, it will eventually turn into a chronic ulcer. Chronic ulcer will eventually lead to cancer, right? So the first thing we wanted to do is to see whether our compounds had anti-inflammatory potential or not. So I screened about 2,000 compounds against, these are macrophages, these are like small cells that we could actually cultivate them in the laboratory. These are called raw 267.4 American type culture collection cells that we bought, and we did this experiment. So we first tested the compounds to see whether they kill these cells or not. If they kill, then they're toxic, I can't use them. But they did not kill. The graph on the left shows that they were not killed. So the viability was very good. So I went on, so I treated the cells with my compounds, and then I put some stress to them using LPS, lipoprotein liposaccharide. I put this stress compound and monitored the pattern. As you can see, the higher the compound that I used, the nitrogen oxide production was low, meaning the compounds were protecting the cells. And then we also studied the protein release factors. Now, when a cell is inflamed, they produce proteins, markers. And based on the markers, we could actually detect whether this particular compound could be used as a drug or not. So we monitored the PGE2 release and also TNF alpha factor. As you can see, for PGE2, with concentration, the NO production reduced. But for TNF alpha, the moment you use a little bit of the compound, it reaches the maximum level, meaning there's a pattern to it. So this compound has potential to be used as drug. Again, we looked at other protein, IL-6 and IL-1 beta. Again, you can see for IL-6, it showed very good inhibition, even at the lowest concentration. By IL-1 beta, just showed concentration dependent. So at the end of the day, we were very sure that the compound that we have stumbled upon had very good activity, and this is a triterpene. I'm not showing the full stereochemistry, so it's been patented, so it's not there's no issue here. And we were able to trace inox, COX-2, beta-actin, some of the proteins that are important as indicators to show that this compound is a potential drug and could be taken to a clinical stage, clinical testing stage. Next, anti-cancer activity. So once we have done inflammation, we wanted to look at cancer. So the best cancer that we wanted to look at was breast cancer, because it is a solid cancer. Solid tumors are easier to work with as opposed to some other cancer, like leukemia and all that. So we wanted to look at breast cancer. But before that, a group of researchers from Kagoshima University approached us, and they were willing to share their adult T-cell leukemia cells with us. So these are called ATL. This particular cancer is very unique. It is only found in Kagoshima and certain part of Hokkaido. Since I studied in Hokkaido and I've got collaborators in Kagoshima, so we're very happy to share some of the cells. And that particular virus that you see is HTLV1 virus. Once the virus enters the person's body, it induces the immune system and it causes cancer in certain organs. And this is only found in certain populations. You and me will not be affected. Only certain population, and the map shows that the areas that are marked pink are the countries that have got this predisposed population. And in Japan, it was Kagoshima and certain part of Hokkaido. So we were able to get some of these cells and we tested them in our lab. And to our surprise, we were able to identify some of the compounds that were very potent towards this disease. As you can see the virus and you can see the person's body, the morphological differences in the person's body differ. Some will have chronic ulcers, some will have pigmentation, some will have this sort of marking in their body. But it's a very serious cancer and it's not very curable at this point. So they were looking for new compounds. And what our study showed that some of these compounds that I've showed from Lawrencia majuscula were very useful. And some of the data that we got, it shows that IC50 was very favorable. And then this compound, the 5 that I show you here, actually cause apoptosis. Apoptosis is the mechanism of killing the cancer cell lines. So it could kill the cancer cell line, and we were also able to study some of the mechanism of enzyme, flip mechanism and all that. So, right? And we were able to study the mechanism of action. Then we went on to study the MCF7, that is the cancer cell lines, breast cancer cell lines. And here we found one compound from soft coral that was able to kill some of the cancer cell lines. And as we moved on with certain concentration, we were able to mark the flip mechanism. We looked at capsase 8 inhibition pathway. These are all scientific jargons, but what I'm trying to say is that our compound was able to connect with the flip mechanism. It was able to trigger certain genes and respond and how to arrest or to how to induce cell death in cancer cell lines. So this compound is very unique, and I think we have patented this compound. Next, this is interesting. Now, when a person has got tumor, primary tumor, when the tumor grows to become a secondary or it migrates. Now, when the tumor grows, angiogenesis takes place. Angiogenesis is the development of blood vessels. So nutrients are taken into the site, cancer cell grows. At the same time, there's another process that's happening. It's called lymphogenesis. That is the lymphatic system. Lymphatic system are similar to blood veins, but these are the systems that are important to assist the flow of bodily fluids. It's just underlying the blood veins. So we wanted to find a compound that could actually inhibit the migration of cancer cell. Now, if the cancer cell migrates through the blood, it will end up in some organ. But if the cancer cells were to migrate through your lymph system, it will be a reservoir in the lymph nodes. So usually people with level 3 or 4 of cancer, it's usually because the cancer cells are in their lymph nodes. So we wanted to avoid that. We wanted to make sure that there's no migration. So I wanted to look at the vascular endothelial growth factor in some of these cells. So as you can see here, the metastasis stage is the migration of cancer cell. Yeah? And if we could stop the migration of the cancer cells into the nodules or lymph nodes. So the lymph nodes are areas where the cancer cell survives, proliferate, and also become dormant. So usually when you send a biopsy to a pathologist, he will look at the lymph node and he will decide how long the person can live. So we wanted to stop the cancer cells from going to the lymph node. So what we did, I worked with some people, some researchers in Carif or in Avanti Cell. They were able to assist us in, you can see the liquid that is flowing, it's actually a lymphatic system, fluorescence lymphatic system. This was induced in GMO, genetically modified zebrafish. We just use the zebrafish. So you can see the graph on the left hand side, lower green color, the black background. This shows the lymphatic system in a zebrafish. Normally, zebrafish will not show you this lymphatic system. These are transgenic zebrafishes. So we got these, we use our compounds against these transgenic zebrafishes, and we treated our compound, and then we looked at most importantly, we wanted to know whether our compounds could stop the lymphatic system from growing, or the lymph system from sprouting. We call it sprouting. So the compounds did not inhibit sprouting or lymphatic system, Now the graph that you see as labeled as SM20 micromolar, this compound is a standard compound used today, and it destroys the sprouting of lymphatic system, but our compound did not. So what we did, we went on further, and this particular compound that I show you, the compound that I show you was interesting because it could actually inhibit the migration of cancer cell, but not the sprouting of the lymph system. So it was very interesting. So in essence, if there is a tumor, you have to introduce this compound, and this compound will arrest the lymphatic system. There will not be migration of cancer cell to other parts of the body or the nodule. Then you can remove that solid cancer. Then you remove the compound or the chemical, and then the cell stays put. So it is a very interesting compound, and the structure of the compound is also very simple. So meaning to say, it could be easily synthesized and made available. The final few slides, I'll talk about the microarray gene expression. What is this? Now, I started off with anti-inflammation. We went on to solid cancer, and then how we knock off the lymphatic system and reduce the migration of cancer cells. Now, again, using the compounds that I've isolated, I was able to show that some of these compounds actually work as triggering mechanism at the gene level. Now, usually, when you do a bioassay, when you use this compound, any drug, it works with the protein that's in your blood. But how are proteins produced? Proteins are produced when your genes are super expressed. So we were able to use this compound to upregulate and downregulate some of the genes. So this study was performed using human primary breast cancer cell lines. What is a human primary breast cancer cell lines? These cell lines are actually isolated from patient. These are first generation cell lines. You can't use the ATCC collection because they are all laboratory cell lines. So we got these cell lines from patients, from doctors who are working with breast cancer patients, and they give it to us. We made sure that these cell lines are not compromised in terms of hormone or chemicals, and then we use them. We've got very stringent process. We use this compound, triterpene, and we look at the activity of the gene. We studied the gene expression, and there's a term that we use, fold change factor. If the expression is up regulation or down regulation to the fold of 1.2, then it is not by chance, it is because of the chemical. So what we did, we set our standards very high. We used 2.0 and 3.0 as the fold change, meaning to say only when the gene expresses itself 200%, only then we accept it as positive. So which is very stringent quality control, and we were able to show that some of these, one of these compound, out of 2000 compound, we were able to show that this compound could actually do this. And again, we did heat map. We wanted to study two-dimensional clustering, whether the up regulation and down regulations were even, whether they are statistically significant, yeah. And then we also did the volcanic plot to show that we are looking at a very narrow window and 95% confident level. We could show that this compound indeed caused up regulation and down regulation of genes, and then these genes were actively producing the proteins that needed to arrest some of these cancer problem in breast cancer patients. Again, we did PCA, principal component analysis, to make sure that up-regulated and down-regulated were clearly separated, and cluster dendrogram to show that there is regulation and the two experiments were not biased. With that, we were able to put it all together and to show that this particular compound, which is a triterpene, was a potent breast cancer, anti-breast cancer compound working at the gene level. And these are the genes that were identified. The red ones were down-regulated, the blue ones were up-regulated. So we went to the level of gene to show which gene causes what activity when we apply our compound. So we look at the drug discovery that I did in my laboratory. We started off with anti-inflammation, and then we went on to cancer cell lines. Then we went on to arrest the lymphatic system to avoid cancer cell lines from migrating. And then we went on to look at the upregulation and downregulation of the genes which are responsible when we use our compound, to show that it's totally safe to use some of this compound, but of course it's subjected to clinical trials. So these are the 5 genes that we have identified. So in conclusion, what my study for the last 21 years has indicated that first, as a researcher, one needs to look at the environment, nature, and we need to learn from nature. When we look at a simple bee coming to a plant, it is not coming because the color is nice. Probably it's coming because there are a lot of VOCs in the air, volatile organic chemicals attracting the bee. Like tuhau, most of us eat tuhau. Tuhau has got a very pungent smell. But do you know that the smell in the tuhau was meant to repel insect from the plant? Because the flower of tuhau, ethylene gera, is at the bottom. And pollination is done by mammals, small rats and mammals and all that. So the chemicals were to repel the insects. Likewise, when you look at nature, we learn a lot of stuff. And it also gives us a glimpse of the evolutionary ecology of certain plants and animals, which are very important. So chemicals that are involved in ecological processes, we call it ecological chemicals, are important chemicals that should be studied, should be understood. And if we were able to show what happens in nature, eventually, when we bring it to lab, we could actually use these chemicals as important starting material for various experiments. As what I've shown, we could use the secondary metabolites as chemotaxonomical markers. We could use them as important nutraceutical. We can use them for anti-inflammation, anti-cancer, even to arrest lymphatic migration of cancer cells through lymphatic system. So the potential of secondary metabolites are fantastic. So to go back on the aim of my study, I think I was able to address both the aims, and the most important one, we could, without any doubt, I could say that there is a link between natural compounds and human health. We can use them to solve our problem of bacterial resistance towards antibiotic. We could solve our problem by using some of these compounds as alternative medicine for certain diseases that we have in our community. With that, I would like to acknowledge the various funders, Kementerian Pendidikan Malaysia, Organisation for the Prohibition of Chemical Weapon, Jeju National Universities, Cancer Research Malaysia, Hokkaido University, SABC, GSPS, IKEA, and few other organizations that have funded my research, and I'm very thankful for that. And this is a photo taken in 2003 with the first batch of my master students in IPMB. At the time I had the whole full head of hair, but not anymore. That was in 2002, 2003 when I was in IPMB. With that, I thank you very much.
Terima kasih Yang Berbahagia Profesor Dr. Charles Vairappan atas syarahan yang disampaikan itu tadi. Sebagai tanda penghargaan dan kenang-kenangan, majlis dengan segala hormatnya menjemput Yang Berbahagia Profesor Dr. Charles Vairappan untuk menyampaikan buku sempena syarahan perdana kepada Timbalan Naib Chancellor UMS Yang Berbahagia Profesor Dr. Rasid Mail. Majlis dengan segala hormatnya mempersilakan. Hadirin sekalian, dengan itu, bagi mewakili pihak Universiti Malaysia Sabah, pihak ahli jawatankuasa pelaksana memohon maaf di atas segala kekurangan dalam majlis pada pagi ini. Bagi mengakhiri majlis ini, dijemput untuk semua berdiri dan bersama-sama menyanyikan lagu Universiti Bertekad Cemerlang. Assalamualaikum warahmatullahi wabarakatuh. Terima kasih.